Thank you. It's been an honor to be associated with ICTS from the beginning, and I have to say, of all the disciplines we've been hearing about at this fantastic meeting, I think biology is one that in particular needs spaces where theory can be protected and where it can collide with other disciplines, and I think ICTS has been exemplary in that aspect. So, and thank you all for staying to the end of this meeting. I hope to tell you in a very short time a lot of different aspects of cell biology. So I have my task cut out for me. This, by the way, is a place called Loki's Castle, 2.5 kilometers below the surface of the ocean, just off the coast of Norway, and I'll explain to you why that's relevant. Okay, so molecular archaeology. What is molecular archaeology? I think the nicest example of this I've seen. The point of molecular archaeology is you have to use the molecules that are information-carrying molecules in biology, which is proteins, RNA, DNA, and somehow ask a historical question that can be solved looking at these molecular sequences. This is a great example where the origin of clothing was diagnosed, the timing of this was diagnosed by looking at the DNA of head and body lice and to see when they diverge. And I think the whole point about this is to show that if you can phrase it as a detective story where the sequences contain the answer, then you can actually get at quite unusual kinds of historical questions. So my goal is to do exactly the same thing, just for a slightly older problem. So you all know, maybe it's in the textbooks, the idea of prokaryotic and eukaryotic cells. Actually, this concept is rather recent. It's only been in the textbooks from the 1960s. And over the last 5 years, there's been a huge renaissance in understanding the origins of eukaryotic cells. Human cells, for example, are eukaryotic in nature. One of the first leaps in our understanding of this was through the work of Carl Woese, who went out into the world and just sampled DNA sequences and looked at how diverse they were. And to his shock and the shock of many other people at that time, turned out that there were previously unknown groups of cellular life on Earth which were not simply visible through their surface phenotypes. And what he realized is that there are actually 3 kinds of groups of organisms on Earth. There's the bacteria, which are like your E. coli and bacillus and so on. There's a group called Archaea, which are another group of unicellular prokaryotic life, but they're not bacteria. And then there's this group, the Eukarya, which includes plants, animals, fungi, amoeba, and so on. And so this 3-kingdom classification of life became the norm. So then enter Lynn Margulis. This is Lynn Margulis on her wedding day with Carl Sagan, who came up with this hypothesis. Well, she refined and really put forward this hypothesis that eukaryotes arose as a discrete event when bacteria engaged in a partnership with some other type of cell. The bacteria eventually became the mitochondria that we now use to make energy inside eukaryotic cells. It's actually quite relevant that Lynn Margulis' original paper was rejected from about 12 different journals before it was published in the Journal of Theoretical Biology in 1967, and we're actually celebrating the 50th anniversary of this influential paper this year. So here's where the work begins. So Loki's Castle, 2.5 kilometers below the surface of the ocean off the coast of Norway. 2 years ago, through submersible experiments, we found collections of DNA which hinted at what the other partner cell was to the bacterium that became the host of the mitochondrion. That is a group which is, in fact, turns out to be an Archaeobacterium. And work that we've done has shown that many of the proteins that's in eukaryotes today actually turn out to be just hybrids of things that were in bacteria and in Archaea. So we are nothing but a combination of bacteria and Archaea. And the trick is then to reconstruct how that crazy event could have happened. It didn't happen in a single flash. It must have taken several millions, tens of millions of years. We don't really know. So here's the broad timeline. So somewhere around 2 billion years ago, there were only prokaryotes on Earth. And then at some point in time, there was a partnership between the bacterium and the archaeal ancestor to make this proto-eukaryotic cell. And that cell, the descendants of those are all the fancy groups of eukaryotes that are living today, including at this end, the animals, which include us, our close cousins, the fungi, plants on this side, and everything in between. So now I want to do molecular archaeology. I want to somehow reconstruct, if we can, to whatever extent we can, what happened over here and how we went from a prokaryotic ancestor to the eukaryotic descendant. And it seems like a crazy question that may not have an answer, but it turns out we can make progress precisely by looking at this molecular information. So remember, you have to use a clever angle, a hook, in order to ask the right question. So one of the questions we asked was, well, if the mitochondrion was a free living bacterium, which lived outside another cell, eventually ended up living inside that cell, somehow the free division of that bacterium must be tamed by the host cell in order to establish a long-term stable partnership. So we said, how do mitochondria divide? And let me explain this very briefly. Bacteria, all bacteria divide by pinching in a sort of purse string manner using a protein called FDSZ, which makes a polymeric ring at the center. Mitochondria used to be bacteria, but they don't divide that way anymore. Mitochondria exist in a host cell, and it turns out the host itself clamps down on mitochondria using another protein encoded in the host nucleus called Dynamin. Dynamin makes a ring on the outside and squeezes the mitochondrion to divide. Interestingly, chloroplasts, which also used to be bacteria, are pinched from the outside by the host, but also pulled from the inside by the original ancestral bacterial machinery. Here's another piece of information which will be relevant later, so I bring it up now. The same topology by which Dynamin pinches membranes and causes mitochondria to divide is also used at the cell surface by eukaryotes to pinch vesicles, which are these sacs that eukaryotes use to eat. That's how they bring food into their interior. And vesicles also get pinched by another copy, another variant of the same protein, Dynamin, by using the same biophysical principles. So the summary of all this is there's a protein called Dynamin, and it regulates the growth and division of mitochondria. And by looking at the evolutionary history of Dynamin, we might say something about the early stages by which mitochondria got integrated into the eukaryotic host cell. So what did the early division apparatus look like? And how do you answer this question? So this is my first bit of theory. You have to ask, what ways can you reconstruct ancestral traits? And in some sense, Darwin already knew how this was done. So in Origin of Species, this figure, by the way, is not from Origin of Species, but it's from his notebooks. It's his first picture of a tree. And the tree is really central to Darwin's ideas of reconstructing ancestral states. So Darwin says, in looking for gradations by which an organ or a molecule or anything, a mitochondrial division, has been perfected, we ought to look exclusively at lineal ancestors. He's saying we should really build a time machine and go back and see what happened. But since this is not possible, we have to look at collateral descendants from the same parent and look for anomalous forms that may almost be called living fossils. So he says the only way to reconstruct the past is to hope for the best and hope some vestige of the past still survives to the present day. Is this possible? Well, it is in some cases, and I'll show you it is in this case. The other little bit of theory that is involved in this talk is the following thing. We're really talking about billions of years in the past. So mitochondria are things that are at least 1.8 to 2 point something billion years old, and we can date this by various means. So a lot of the information that we need to do this reconstruction has been degraded. And in order to look back, you have to use some clever tricks of data analysis. Okay? And roughly the clever trick involves the following thing. This is a famous portrait by Dali. And the point about this portrait is to show that if you really squint and step back, you realize it is a portrait of Abraham Lincoln. It's a story about asymptotics. It's a story that you can't look at the details of an image after this much time has passed, but you need to somehow coarse grain. And let me explain how we do that. So this is not just abstract, there's a real way we can do this. Here's the general idea. There is a tree, and this is a tree of life. It's a tree of species, which says these species have descended from a common ancestor through various branching events over the course of, say, 2 billion years. And in the present day, we find protein sequences that exist in the genomes of these species. These are the real protein sequences of that Dynamin protein I described to you from different species. And now we want to reconstruct the ancestral state which doesn't exist anymore. And one way to do that is just to line these up as well as we can. And then we can say that, well, if there's some sort of common amino acid, these are all amino acid letters, by the way, common amino acid at the same site in the living proteins, then maybe the ancestor also has that amino acid in that position. And you might be right, but it might also be accidental convergence, because amino acids change into one another all the time. And there's another problem, by the way. It could be that amino acids have diverged so much that you can't at all guess what was in the ancestral state. The information has just been overwritten. Okay, so by looking at the level of single amino acids, you cannot really make any progress, and you have to coarse grain somehow, just like in that portrait of Lincoln. So you bring in this idea of coarse graining. It is rather simple. You simply break up the protein into chunks of some length of amino acids. You can pick it arbitrarily. At some length, you start to find that there are common sort of patterns of amino acids that are present in this variant of the protein in humans, and in that variant of the protein in plants. I've just colored this purple arbitrarily. And you might guess that the ancestor also has a purple variant at that location, meaning it's similar to the two present-day variants. At some other position, the variants have diverged so much that I haven't labeled them by the same color, you can't really guess what the ancestor had. So these coarse-grained positions, which you could almost call fake amino acids, these are just single letters that represent larger clusters of sequences. That's the level at which to analyze sequence evolution at very long timescales. By the way, if you make these lengths too long, then you'll find that no two proteins are identical, and by definition, you can't reconstruct the path. So this is indeed an asymptotic analysis where single amino acids, there's no information preserved. very large stretches of proteins. By definition, no two things are alike, but at an optimal chunking of the protein, you can actually reconstruct the past. So from now on, for the rest of the talk, I will represent protein sequences, but each letter over there actually represents a stretch of amino acids through some sort of dictionary rather than a single amino acid. So in fact, here's the Dynamin protein. It has 3 domains, and that doesn't really matter for the purposes of this talk. But instead of looking at the sort of physical structure of Dynamin, you just stretch out the 600 amino acids of Dynamin along a matrix, and then you ask a question about coevolution of different sites, and you use that to reveal what the optimal level of chunking actually is. So the protein itself, if you probe, it reveals the lengths at which you can consider these fake amino acids as carrying important information about the past. So in fact, it's very, very analogous to the idea that you step back at a certain level, which varies anywhere between sort of 10 and 100 amino acids. And you can do this for any protein that you want. Having done this, you realize that in this particular case, there's about 8 different chunks. And so you split up the Dynamin protein into these 8 different fake letters. And then you carry out the following very important task. So this is a very common trick in phylogenetics and bioinformatics. And let me try and explain how that's done over here. It uses a principle of parsimony, but in a very specific sense. It says if 2 organisms in the present day share a complex trait, and you can rule out convergence of that complex trait, then you can infer that the ancestor had the same trait. Okay, so there's a lot of assumptions built in here. In other words, if we can find long enough chunks that there's no way 2 proteins could have had the same amino acids for the entire stretch through convergence, then you can assume that the ancestor had that trait. This is called Dollo parsimony, and in this way you sort of kick back that amino acid to older and older and older species that must have existed in the past. So let me walk you through this again. This is the tree of species that I've always been describing. Here's an ancestral species. Here are 4 of its present-day descendants. That's the tree of species. You can make another tree, and this tree is fundamentally different. It is a tree of proteins. It's your standard phylogenetic tree. It just tells you how pairs of protein sequences are related to each other. That's this diagram over here, and let me walk you through that. This, for example, are the sequences at some particular chunk in the Dynamin protein, say very close to one end. In this particular case, there are maybe 3 flavors of letters that you get at that position. And what I've drawn here is very much like Darwin's diagram, where extant species, species that are still alive today, I've colored with filled circles, and extinct species or extinct versions of proteins I've colored with empty circles. And proteins diverge. Protein keeps changing over time. Protein is selected, or it's just random variation and fixation of the population. So this thing just keeps on diverging. Now, what I want you to note is the following thing. This extinct version of the protein, that version no longer exists, but there are plenty of present-day organisms that contain versions that are close to the ancestral state. As long as you find those organisms, you can have a reasonable guess what the ancestral state was. So it's important that you have these clusters. But there's another thing. Because chunks have diverged, there are many present-day organisms that contain protein variants that have nothing to do with the ancestor. By actually overlaying this kind of data, by giving names to these clusters and placing it at the leaves of the species tree, you can play the very simple game. You can teach it in school. If you see the same letter at 2 positions, it must have been present in the past. If you see the same letter in very restricted subsets, it's present more recently. It's also possible that no present-day version of a protein exists today, in which case you've simply lost contact with the past. It's just not possible. And so by putting the gene tree on top of the species tree, you can do ancestral reconstruction. This is all very simple. This is exactly how it's done. You can do it on pen and paper. Theory doesn't have to be hard in that sense. So this is actual data. It's meant to mimic this abstract figure over here. The representation over here is something called a phylogenetic network rather than a phylogenetic tree, which preserves senses of uncertainty present in the data, and I don't want to get into it right now. But the point is, I can play exactly the same game. I can find clusters of letters. I can place the letters at the base of a tree of the various groups of eukaryotes present today, like animals, fungi, plants, and so on. And I can I can sort of roll back the clock to see what was present in this thing which we affectionately call LICA, which is the last eukaryotic common ancestor. Okay, so now that I've just shown you in the principle of how things are done, let me just show you what we actually find. So this is an actual time frame, and this is the evolution of the Dynamin protein across literally 1.8 billion years of life on Earth. And I'm glad that the astrophysicists and the biologists can both say billions of years. I think this is actually really exciting that from either by looking really far in the sky or by looking really deep in molecular sequences, you can actually push back the clock over billions of years. It's something that can actually be done. So it's complicated, but it's exactly the same as the abstract model I showed you earlier. These are species trees. These are versions of eukaryotes that are present today. These are the sequences in this funny notation of that mitochondrial division protein that's present in each of these organisms. And by pushing back letters wherever they are common, I can reconstruct an ancestral state. Okay, I can actually do this for many variants of Dynamin. I've done it for the Dynamin that divides mitochondria. I can do it for the Dynamin that divides vesicles. They actually share letters in certain positions, which means even these two proteins must have come from a common ancestor, okay, in ways that are reasonably complicated. And it's using this kind of data that you're going to have to answer a much more specific question of what the ancestral machinery looked like. It's done using the entire complicated data set, but let me just summarize it for you over here. Once again, I have, in this case, a schematic species tree. Animals, fungi, green algae, other kinds of things, alveolates, these things live in the ocean. There's all the eukaryotic diversity is over here. Time on the x-axis. The label now along the tree represents our inference of what the mitochondrial division machinery looked like in that ancestral species. And it is the following. If you see this sort of dotted or dashed lavender and blue line, it means that we reconstruct in that species that the Dynamin system looks like this, where, maybe not surprisingly, it contains only one copy of Dynamin, and that version of the protein does many things in the cell. In particular, it divides mitochondria. It also divides vesicles that the cell uses to eat, and it's exactly the same protein that does both. There are, however, other variants of Dynamin, and that you see over here in our lineage, also in the lineage of plants and in alveolates. In this case, as often happens in genomes, the gene encoding Dynamin duplicated in the genome. And then what happened was one copy of that gene took over one specialized task, the other copy took over the other specialized task. It's not surprising that they diverge because you've released the constraint of having to do a much more complicated job. So what happens in our cells and in plants is that you have one version of Dynamin that pinches mitochondria, you have another version of Dynamin that pinches vesicles, and they do their own thing. And importantly, please notice, we guess that the ancestral state of the mitochondrion also had the bacterial division machinery because they used to be bacterial. But in our cells, mitochondria divide purely through external means. Now, turns out that's not the only type of eukaryote that survives to the present day. There is another type, and it's exactly the other variants of this tree that I've shown. And I'll just flash back and forth so you can see the difference. The point is, this is the derived state that humans and plants and so on use. And this one is not a reconstruction. It's really the present day state of the mitochondrial division apparatus. It turns out to be identical to the reconstructed ancestral state. There are, in a few cases, in fact, many sort of strange organisms that people don't study in the lab, stramenopiles, amoebozoans, red algae, creatures which contain the exact same Dynamin sequence as the one we reconstructed as the 2 billion-year-old version. Okay? And it turns out when you read the papers about how these things divide, it's the same Dynamin that does both things. So this is not a tautology. I'm saying that A, we reconstructed that the Dynamin looks ancestral, and B, cell biological studies have shown that that Dynamin actually does both jobs simultaneously. So what have we established? We've actually established something reasonably interesting, which is by doing an ancestral construction of a sequence, you find some ancestral putative 2 billion-year-old cell, and you don't have to believe me that that's the right ancestral reconstruction. I could have been wrong, my statistics could be noisy, and so on. But we gain more confidence because we find that that predicted ancestral sequence, if we troll through the thousands of genomes that have been sequenced today, that ancestral sequence is still present in a few organisms. That's step one. Now you go to those few organisms, and luckily a few people have done the experiment. And they've pulled out those few organisms, and then it turns out that those few organisms use a division machinery that is very similar to what you would have expected the ancestral version to be. So this is a strong suggestion that these creatures, at least their mitochondrial division apparatus, is the type of living fossil that Darwin envisaged. In other words, we don't have to build a time machine. For some reason, some creatures today have preserved the mitochondrial division apparatus in a way that hasn't changed in 2 billion years. And if you want to study that, you should go and pick up these creatures. Now, the important point about this to me is it goes beyond just the idea that we reconstructed the mitochondrial division system, although that's very important for the history of eukaryotes and to Lynn Margulies' hypothesis. I think it actually says something more about the way biology is done today. So today in biology, we live under the tyranny of model organisms. If you don't work on the fly or the worm or yeast, or human, it means you're not doing anything at all. And this absolutely throws away the most important aspect of biology, which Christos highlighted in the first talk today, which is the diversity of biology. Biology is the study of diversity. And if you throw out diversity, you're missing everything. But diversity is also very difficult to work with. And what I think these kinds of molecular archaeology efforts do is they allow a researcher to pick an appropriate organism as a study subject from the list of all the ones that have been sequenced today, bring it into the lab and make it a new model. And with tricks and techniques like CRISPR, this kind of thing has never been as easy as it is today. So at least for the students out there, the call to arms is the following thing. Do not work on any organisms that people have already been working on. And I think Sri also said today, don't work on subjects that are overcrowded. Pick new organisms, pick new systems. And if you don't know where to pick, go look at their genomes and find something interesting that the genome can tell you and use that as a way to select. Okay, so I'm going to wrap up with a few open questions in eukaryote evolution. And these are all the other questions that we study today. So I told you about the following thing. So somewhere, so this is a timeline. This is billions of years ago. That's the present day. And in the present day, here we are, animals, plants, amoeba, and all the eukaryotic cells that survive on Earth today. The really surprising fact is that all extant eukaryotes are nearly identical to one another on many parameters. They all have nuclei. They all have microtubules. They all have mitochondria. They all have a Golgi apparatus. And so, and they all do sexual reproduction. They all do mitosis. So somehow, there's been a huge change from a prokaryotic ancestor to the present-day eukaryotic state, and all that diversity during the change has vanished. Okay, it's gone. And if we want to reconstruct, maybe it's surviving in some deep pocket in the ocean, and if so, we'd be lucky. But otherwise, we have to reconstruct it just by imagining what happened during that state, during that transition. So the timeline for this is we know that there was no oxygen on Earth before 2.5, no free oxygen, at least in the atmosphere, before 2.5 billion years ago. Photosynthesis is invented, oxygen comes into the atmosphere, aerobic bacteria arise, the aerobic bacterium becomes the mitochondrion of a proto-eukaryotic cell, something happens. And somehow, between that proto-eukaryote, which looked very prokaryotic in its structure, you get this very complicated present-day eukaryotic cell. How do you go after this question when you can't reconstruct ancestral cellular states? The way you go after it is because, coincidentally or causally, one of the other things that happened to eukaryotes is that their genomes increased in size by a factor of 1,000. Okay? And that meant that individual gene copies were present in multiple copies, and eukaryotes also invented new types of genes to carry out all their functions. And because we're so good at protein sequence reconstruction and phylogenetics, we can actually use this informational layer to make predictions about what happens at the cellular layer, and in that way, reconstruct what intermediate ancestral cells were like. So I have a couple of minutes left, but I'm happy to take questions. So I'll close with that. A lot of this work has been written up. The work on Dynamin was done by my graduate student Ramya. And a lot of the other work that we do in the lab now is not just to go after ancient eukaryotic evolution, but to understand that evolution is still proceeding today as fast as it has always been. And in fact, you can recapitulate many of the predicted changes that could have happened in the past in the lab. And I think that's a very exciting area of biology that people should be getting into. So with that, I'll stop, and I'll be happy to take questions. Thank you.
Questions? Comments?
Mukund, I gather, I presume one of the reasons you studied mitochondria is, one, they're very critical, happened early, but also because they asexually reproduce, so it's infinitely easier to construct these trees. Is it really possible to go back 2.5 billion years when you, sexual reproduction?
Okay, thank you. So it's, by the way, the Dynamin protein is encoded in the nucleus. Okay, it's not part of the bacterial mitochondrial repertoire, but in fact is inherited sexually through the host tree and is swapped. And by the way, I didn't show it to you, but our trees do show that genes can jump from one branch to another, and that complexity exists. Okay, it can make it messy, but we can deal with it. Yeah.
Mukund, the nucleus didn't enter directly into your story.
No, it didn't.
It's almost as though mitochondria are what make your eukaryotes different from prokaryotes. So where is the nucleus?
Where's the nucleus? So Buzz Baum published a fairly controversial theory about the origin of the nucleus. Let me tell you, the eukaryotic evolution field confronts this fact all the time, that everybody has their favorite parameter for what makes a eukaryote, but they coincide on every cell. Okay, we all have mitochondria, we all have nuclei. So any story you can make at that point is sort of fundamentally not testable at a cellular level. There are theories that because you had mitochondria and you had this extra genomic material, you had to protect the host genome. But I don't quite believe that. The better theory is that the very entry of the mitochondrion topologically created another compartment. It's a sort of mathematical consequence, and that simply is what we see as let me put it very simply. The archaeal membrane is the nuclear membrane, and everything else is a process. Okay? But I'm happy to talk more about this later. There's no evidence one way or the other for this. Yeah.
So that was a fascinating talk, and I asked this question as a physicist, and I find biology incredibly complex. Just learning the language seems to be hard for me. So this question may be ignorant or it may be profound, I don't know. When you do these trees and trace them, you find some examples of living fossils, and you had 3 of them up there. Can you is it an important question to even ask why they survived? Do they live in environments that are just totally unchanging, or is there something else going on?
Okay, so it's let me go back to this tree. So here's the fact, and if I spent more time with the raw data, I could explain this to you correctly. At every scale, evolution is heterogeneous. The level of the whole organism, some organs evolve faster than others. Some things that are suited to their environment stop evolving. At the level of a single protein, one end can evolve while the other one stays still. Okay, so at one answer to your question, it's a pure sampling issue. If you look through enough, some bits and pieces of enough things would have survived. There is a better answer, which is that there are layers of buffering in cells, and to some extent the genetic code is a living fossil because it is totally buffered from any change in the outside. If you've been freed from the pressures of selection, you have no reason to change, especially once you get linked. But I would say it's a surprise when you do see a living fossil rather than the other way around. Yeah,
there was a report, about 2 years ago about a finding of a very ancient eukaryote, and I just wonder whether you'd make some comments about that, and whether is that holding up, and is that something that is a remnant of that that proto eukaryotic cell that you showed up there.
So before I answer your question, every cell today is as old as every other cell today. Okay, so to say that things are remnants mean that we have to prove that certain aspects of them are living fossils. When you say there was an ancient eukaryote, there are 2 things that happened. One is they found fossils of ancient algal mats, which date back to something like 1.2 to 1.5 billion years. The other thing that happened was the discovery of this creature, which is Loki Archaeota, which was not a eukaryote. It was a prokaryote whose genes are as close to our genes as perhaps 2 eukaryotic genomes are today. So all this says is that we found our closest cousin who is not a eukaryote. Now, unfortunately, this thing lives at 2.5 kilometers below the surface of the Earth, and so we haven't seen it. And one of the lovely things in biology today is we have genomes of organisms that we just cannot culture. And it's a nice mystery to reconstruct what their biology is like. So this is that creature you're talking about. Okay. Oh.
Okay.
I think there's one,
first let's,
Gautam had a...
Is there a question?
Mitochondria make their energy through this
Yeah.
ATP synthase.
Yeah.
I was just wondering, where does that come from? Did that come from the Archaea? Did that come from somewhere else?
So humans contain many ATP synthases, but in particular they contain one ATP synthase on the mitochondrial membrane, which comes from bacteria. They contain another ATP synthase on the outer membrane, which comes from Archaea, which sort of fits the bill. The outer ATP synthase does many things, but it maintains the membrane potential on the outside of the cell. And that whole story hangs together, except for one funny thing. The lipids on the outer membrane ought to be archaeal lipids, but instead they're bacterial lipids, and that's another crazy mystery, and we don't understand it. Right? I don't. Okay.
Well, first let's thank the speaker.